This is lecture 13, part 2. We're going to begin our defining process of color using a systematic approach and get into some of the industrial aspects of color science. So just as a review, in the part 1 of this lecture, I defined or stated that color, or what we perceive as color, the stimulus, has its roots in psychophysics, and we can try to make instruments replicate that experience. The stimulus itself is a combination of three contributing factors, namely a light source, which has its own power distribution or spectral distribution, the object that interacts with light and either absorbs or reflects light according to different wavelengths, and then a detector, namely the observer, which has its own unique response function, namely what wavelengths it sees preferentially. So the human eye as opposed to some of the photovoltaic type devices. I'm going to put off the detectors until instrumental analysis. So I'll describe how a photomultiplier tube and a silicon diode detector work in instrumental. In part 2 and 3 of this series in quantitative analysis, I just want to go into a more quantitative, shall we say, definition of color and use that as an introduction to absorption analysis, but at the same time introduce you to a branch of analytical chemistry that's used in industry. And we'll be drawing on a few classic textbooks, one of which is The Color Kittens, which is one of Domino's favorite bedtime storybooks. We'll need to make a few definitions, starting with the two defining terms, namely achromatic items or objects and chromatic objects. So the achromatics have no color at all, or what we perceive as color. They are either white, black, or some shade of gray. We'll define it what this means in terms of the spectral distribution shortly. And these are distinguished from the chromatic objects, which have an attribute that we would define as having a color. And there are specialized terms in relating or defining color, and one refers to the hue, H-U-E, as a specific type of color. And we'll start using definitions from Bill Meyer's classic textbook. So his definition of hue is given at the bottom. Pretty much every single one of the classic textbooks that I have consulted for these lectures has the following exercise as a kind of thought experiment and outlining the approach of how one makes a definition of a color and a systematic way to define color. So they talk of this as the desert island or the pebble sorting exercise. So you begin by having a collection of pebbles of different colors, and they'll be sorted into categories. So the first task naturally would be to remove the achromatic pebbles from the pile and sort them according to their lightness or their grayscale, which we'll talk about shortly. Then you would identify and select the pebbles of a specific color, say yellow, and gather all of those together. Then you would go for blue, red, and so on, and then so in other words, create piles of pebbles according to their color, or as we call them, the hue. One can then take the piles and sort them by how light or dark they are. And in the case of the achromatics, that sorting is referred to as lightness. In the case of the chromatics, it's referred to as value. So the terms differ, but we can attribute value to the shade or the lightness or darkness of the object. And then finally, we recognize that there's differences in what we call saturation, how intense the color seems to us. I'll talk about this in a more formal way in the next slide. And we call that characteristic the chroma. So just as a review, we have hue, we have value, and we have chroma. So looking at the formal definitions of these words now, according to Billmeyer in his 1981 textbook, hue is the quality of the color, value is the lightness of the color, and you can relate this to the grayscale, and chroma is the intensity of the color, in other words, the saturation of the colorfulness. And most of the textbooks give the example at the bottom to illustrate the chroma effect. So if you have a brick and a tomato, they both have the same hue. They're both could be called red. They can have the same value. They can have the same darkness when they're put side by side. However, the impact, the stimulus that you get when you look at a brick as opposed to a tomato is markedly different. And the photographers or painters would refer to this as saturation of the color. In scientific terms, we refer to this as the chroma. If we look at the sorting exercise relating to the achromatics, we develop what we would call, or some people call, a grayscale, namely a transition in steps from pure white to pure black. And one can define the steps according to the amount of pigment. I'm going to, in the next slide, refer to a grayscale scheme by Willem Ostwald, who was a founder of analytical chemistry and at the same time also color science. Ostwald liked making analogies between color and the musical scales of tones, so he created octaves. And he found that he could pretty much characterize any color or lightness using a scale of 8, and did so. So you'll see this a lot. If you happen to work in the area of photography and work with film speeds or shutter speeds or apertures through the lens, you'll also find that things work in series of 8s, in which each step corresponds to a halving of the amount of light that's being transmitted or reflected from a surface. So a white object reflects 100% of the incident light back to the observer. If it is black, it absorbs all the light, and you see 0 of the light reflected. Similarly, if it is a transparent object, you get 100% through or 0% through. Gray is an intermediary. And the steps taken, and you'll see this in the absorption when we talk about absorbance, is that the designated steps work in steps of or increments of 1/2 of the amount of transmitted reflective light with each step. And I'll outline this in a little more detail in the next slide. So if we look at a typical photographic lens, so this is a camera shutter with the front of the lens removed, and you see on the left the opening through which the light has to pass. So this represents 100% of the area, allowing the light to pass through the lens onto the film or the charge transfer device that would ordinarily couple to a digital camera. The diaphragm is not visible on the left-hand image. In the right-hand image labeled 1, I have the aperture closed to the first position, so this is a starting point in closing the diaphragm and admitting or controlling the amount of light that passes through the lens. If I move to the second stop, you'll see that the diaphragm is closed in such a way that 1/2 of the original area available is now gone. So the light can only pass through 1/2 of the original. If I go another stop, I reduce it by 1/2 again. So what I'm doing is decreasing the amount of light that can pass through the lens by 1/2 every time I click my diaphragm closed. And so these are referred to as f-stops on the camera. If you are familiar with particularly film photography cameras, you'll also notice that the shutter speeds go in increments of 2 as well. So the shutter speed doubles with each click, allowing half the light to pass through the lens or through the shutter at a given time. Similarly, if you're familiar with the film speed index code, they also work in increments of 2. So the concept of doubling the amount of light interacting with the film or the charge-coupled transfer device that's used to do the digital image is retained very much in the model of Ostwald's convention. So if we look at a spectrum, the spectral distribution of an observed object, we can see the characteristics of the different colors. So we'll skip over how this is done and save this for a later lecture and also for instrumental analysis. But for the time being, we just look at these spectra and look at how they are associated with different types of colors or stimuli that we perceive as color. So in the center vertical column, we have white to black, and we see that the spectral distribution is uniform. In other words, it's pretty much a flat line across all the wavelengths in the visible spectrum. What changes in this situation in going from black to white is the quantity of the light being reflected. So in the case of black, 0 light is reflected or transmitted in the case of an object that transmits light. In the case of white, 100% or very nearly 100% of the light is reflected towards the observer, and gray is some intermediary between the white and the black. So some fraction of the light is being reflected or transmitted. The next column to look at would be the left column, which is the transition from red to yellow. So red and yellow are primary colors, as we'll discuss in another slide to come. But red and yellow have essentially a knee-shaped spectral distribution. In other words, the wavelengths that strike you as the observer are selected according to by that color. So there's a lot of long wavelength stuff coming and very little in the short wavelength. As you move from red to yellow, however, the shift is to shorter wavelengths. And then lastly, if you look at the right-hand column, you have the spectrum of blue in the center. And blue is a third primary color. And if you start to combine blue with red, you get purple. And if you combine blue with green, you get different shades of green. So green being another of the primary colors complementary to red. So if you move away from blue in the direction of red or green, you get different shapes of the spectrum. And notice that they have peak-like qualities. So you're starting with the purple to get 2 peaks, and with the case of green, you're just sort of shifting greens to longer wavelengths. I just want to take another look and describe to you another version of the grayscale, or the transition from white to black, using gray filters. So on the left is a spectrum, transmission or absorption spectrum, of what's called a neutral density filter, which is essentially a gelatin or a medium that is filled with a black pigment and thereby has a universal background absorbing all wavelengths of light. And you'll notice that the spectrum is very flat, except at the point where you start to reach around 700 nanometers. And the reason for that little jump at 700 nanometers is probably due to the physical size of the pigment, which is at this point starting to be commensurate with the wavelength of the light. And so that would start to increase scattering. But the important thing to realize is that the spectrum, the amount of the light transmitted, is really quite flat. And just as a prelude to talking about absorption, note that the optical density is synonymous with the term absorbance that you'd be familiar with Beer's law. and that a 0.3 optical density corresponds to a reduction in the transmitted light by 50%, so that each increment of 0.3 in the optical density, or the absorbance, corresponds to an additional 50% reduction in the transmission of the light. In general, you can buy Ratten filters. This is a type of filter available from Kodak, which are created using pigments synthesized by Kodak and matching idealized colors so that they can be used as standards. So these are very popular in the photography industry, the film industry, and so on, for standardizing the films, for creating effects, and so on. So there's, in the bottom right, there's an illustration of some Ratten filters. You buy them, they're little pieces of gelatin, they're very thin films. The black one is probably a neutral density filter, and you can buy them in the primary colors. And the spectrum, which I found on the web somewhere, of 3 of the primary color Ratten filters is indicated to the left. And you can see that the blue primarily transmit lower wavelength, the green intermediary wavelengths, and the red the long wavelengths. Here I've gone to the original Ratten data pages. The citation is at the bottom of the slide. And I've picked yellow and blue as a comparison to show you the complementarity of the colors, which will be described in a little bit. So on the left is the yellow filter, and these are absorbance spectrums. The previous slide had transmittance spectrum. So yellow transmits almost 100% of the light above 550 and cuts off significantly any light below 550 nanometers. Turning to the blue cuts off much of the light above 450, lets through a band between roughly 400 and I guess 475 there, and then cuts off the lower wavelengths again. If I look at a second complementary pair, namely red and green Ratten filters, I see that red transmits almost 100% of the light above 600 nanometers and absorbs pretty much everything else, whereas green, again, cuts off everything above 600, lets through a little band in the range of 500, so somewhere roughly 520 and 580, I'm calling that. So they are complementary in the sense that one cuts off what the other allows. And we're going to see the psychophysics of this complementarity coming up. The origin of complementary colors is actually a psychophysics phenomenon regarding after images. So according to Ostwald and those who are involved in psychophysics, if you stare at the yellow square for 20 seconds and it disappears, you should see a blue after image, which is the criterion, the initial criterion for complementarity. So start looking at the yellow square. Stare, stare, stare. I don't know, Domino, did you see a blue square? And since blue is complementary to yellow, when the blue square disappears, you should see a yellow after image starting now. Maybe. Anyway, that was exciting. It's likely that you do not believe our little psychophysics experiment, and so there are other experimental demonstrations of the complementarity of colors and the criterion for establishing complementarity. So you have a disk of the two colors, and you can spin the disk. And if the colors are complementary, spinning the colors at high speed or the disk at high speed will yield to your eye a neutral gray or achromic stimulus. And the shade of the gray created depends on the relative areas on the disk of yellow and blue. And since red and green are also a complementary pair, spinning the disk with combination of red and green areas will likewise give you a gray pattern. Again, the shade of gray will be determined by the amount of relative areas of the red and the green. And I should point out to you that if you combine actual pigments, namely reds and green pigments, they do not combine to give you gray. The definition of two complementary color pairs allows us to create a foundation for organizing definitions of colors. So we can imagine a white background and organize coordinate axes using the complementary colors, namely red and green on the horizontal axis and blue and yellow on the vertical axis. One can now work along the quadrants to define additional hues by combining the primary colors. So one can work along the bottom right quadrant by combining red and yellow, the upper right quadrant by combining red and blue, the upper left quadrant by combining green and blue, and the lower left quadrant by combining green and yellow. And so by combining the adjacent colors and mixing them, we can fill in the space, the off axes, in other words, and define new hues in this manner. This leaves gaps that we can fill in a similar fashion. So we can make yellow redder or greener, and so work up from the bottom along the lower left and lower right quadrants. We can make red yellower or bluer and work along the lower right and upper right quadrants by mixing those colors. We can make blue redder or greener similarly. We can make green bluer or yellower. And so the spaces that I have left blank on this little color wheel can be filled in and given specific names according to the proportions to which we mix the primary colors. And this concludes the part 2 of lecture 13. We're going to get into color mixing next, and if you have a cat, you're familiar with this picture.